Cellular respiration is defined as the controlled release of energy from organic compounds in cells to form ATP. The goal of cell respiration is to make ATP. One of the fundamental pieces in cell respiration is redox reactions, which are the energy-releasing reactions. In these reactions, it is easiest to follow the hydrogens. When something is being oxidized, there is a loss of electrons or a loss of hydrogen. When something is reduced, there is a gain of electrons or a gain of hydrogen. A common acronym to remember this is oil rig. Oxidation is loss, reduction is gain. The first step for all kinds of cell respiration is glycolysis, which is the breakdown of one glucose molecule into 2-pyruvate with a small yield of ATP. This takes place in the cytoplasm and does not require oxygen. First, phosphorylation of glucose occurs, and 2 phosphate molecules are added to the end of the glucose molecule. Then, lysis occurs, which splits the glucose molecule into 2 3-carbon molecules. After that, phosphorylation of the 3-carbon sugar occurs, adding a phosphate to the end of each molecule. Next, oxidation of the 3-carbon sugar and reduction of NAD+ happens simultaneously. 2 NADH plus H+ molecules are made. Finally, substrate-level phosphorylation occurs when each 3-carbon molecule gives up a phosphate. ADP becomes ATP, and the new molecule formed is pyruvate. In the end, there is a total yield of 2 pyruvate, 2 ATP, and 2 NADH for 1 glucose. From glycolysis, the pyruvate molecules enter into one of 2 pathways, anaerobic respiration or aerobic respiration. In anaerobic respiration, fermentation occurs. There can either be alcoholic or lactic acid fermentation as a result of this. There is no oxygen needed for this type of respiration. If oxygen is present, then aerobic respiration will occur. The first step after glycolysis is the link reaction, which functions to make electron carriers. First, the pyruvate molecule is decarboxylated. Then the remaining 2-carbon molecule or acetyl group combines with coenzyme A to make acetyl-CoA. Oxidation of the molecule and reduction of NAD+ occur, and NADH plus H+ is formed. After the link reaction comes the Krebs cycle, which occurs in the matrix of the mitochondria. First, the acetyl-CoA molecule combines with a 4-carbon molecule to produce citric acid. The coenzyme A is then recycled. The citric acid molecule will be decarboxylated and oxidized, and NAD+ is reduced to produce CO2 and NADH. The same thing happens with the 5-carbon molecule, producing more CO2 and NADH. ADP is also phosphorylized to form ATP. The 4-carbon goes through more oxidation reactions, and FAD and NAD+ are reduced to produce FADH2 and NADH. The 4-carbon is now back in its original form and ready to combine with the next acetyl-CoA molecule. 1 acetyl-CoA molecule makes 2 CO2, 3 NADH plus H+ molecules, 1 FADH2, and 1 ATP. For every glucose molecule, these numbers double. The NADH and FADH2 molecules from the Krebs cycle then move to the electron transport chain. This occurs on the cristae of the mitochondria, where the proteins are found that facilitate the electron transport chain. First, the NADH plus H+ molecule is oxidized into NAD+. The electron lost is then taken up by another electron carrier. The H+ ion is pumped from the matrix to the inner membrane space. This happens over and over until the electron is picked up by oxygen and converted into water. The releasing of the H+ ions causes a concentration gradient in the inner membrane space, so the H+ ions want to come back into the matrix. However, they can only get back through a special protein port called ATP synthase. When the H+ ions go through this port, ADP is phosphorylized to become ATP. This process produces about 34 ATP. So throughout this process, many products are made in each of the steps. In glycolysis, 2 pyruvate, 2 NADH plus 2 H+, and 2 ATP are made. In the Krebs cycle, 2 ATP, 4 CO2, 6 NADH plus 6 H+, 2 FADH2 are made. In the link reaction, 2 acetyl-CoA, 2 CO2, 2 NADH plus 2 H+ are made, And finally, in the electron transport chain, there are about 34 ATP, 1 ADH plus H+, 1 FADH2 molecule, and water made.